Luckily, I'm not head of Institute of Ecology and Evolution right now. I will be next year, but I'm not now. I'd like to start thanking the organizers for putting this excellent symposium together and having us come together in a very nice place and have very good discussions. So, let's see how this works. Just trying to find out where the pointer is on this. Oh, that's a separate pointer, sorry. Okay, great, thank you. So I'm asking the question today here that sort of follows or sort of relates to the previous talk by Nicola, whether sometimes perhaps species may actually collapse, whether species diversity may actually be eroded by hybridization between species, and whether humans may actually change environments in a way that facilitates such a mechanism of loss of species diversity. Probably my pet organisms that I think I have some evidence for this actually happening at a pretty large scale is the cichlid fish in the East African Great Lakes. So this background here on the slide has various different phenotypes of Lake Victoria cichlid fish. I've been working on these for quite a few years, but I'll talk very little about these today and try to focus on systems that are closer to the Mediterranean, if you like. So a little bit of background. Species diversity obviously can be lost through a variety of different mechanisms, but maybe very roughly one can categorize them as either demographic decline or perhaps merging of species, not necessarily associated with any demographic decline or possibly the two interacting. Introgressive hybridization is often discussed in conservation biology, but it's most often considered in the context of secondary contact between previously allopatric species due to anthropogenically driven translocations. Another way by which humans can affect gene flow and introgressive hybridization between species is through changing the ecological structure of habitats. And this may affect species that have a history of sympatry as much as species that have a history of allopatry. This may particularly often happen when people make originally heterogeneous environments with lots of ecological dimensionality when people make them lower dimensional. And there's many cases where, of course, we know where that's what happens with ecosystems. Now, in principle, as long as any two species haven't evolved nearly complete intrinsic post-zygotic isolation, then anything can happen to their mechanisms of reproductive isolation that would make them collapse back into a single hybrid swarm. And this probably applies to a fairly large fraction of species diversity. So probably a fairly large fraction of species has actually some closely related species that are sufficiently close for them to potentially merge through the breakdown or the meltdown of reproductive isolation. And ever since Coyne and Orr's classical work on the clock-like evolution of post-zygotic isolation, people have repeated this sort of approach, and there's quite a few data now for vertebrates that suggest that there's a fairly large window of opportunity in terms of the time after lineages began to diverge, during which such merging of species is possible because intrinsic post-zygotic isolation is highly incomplete. Somewhere between most of the vertebrate studies that I'm aware of converge on roughly 4 to 8 million years of divergence time prior to the near completion of the post-zygotic, intrinsic post-zygotic isolation. And so almost all the diversity of cichlid fish in African lakes would fall into that window. So Lake Victoria, which is right up here, it's the lake I've been working on most of my career, really has got about 500 different species of cichlid fish that we know of, there may be more, but they all evolved within just a couple of tens of thousands of years. Certainly not more than 200,000 years at very maximum. So all of these species, and they've all emerged from pretty much a monophyletic ancestral population. So there's no intrinsic post-zygotic isolation that we could measure or that anyone else has been able to measure yet. And the same is probably true for Malawi, even though this is quite a bit older. These species still have no intrinsic post-zygotic isolation. And even like Tanganyika is the oldest of these radiations, somewhere between 5 and 20 million years old, a very significant fraction of species diversity within this is actually so young that they would still be able to merge. But this is not something that's necessarily specific for these large, iconic examples of adaptive radiations. There's many other examples of species that are sympatric or parapatric and are maintained by divergent natural selection between environments without intrinsic or without strong intrinsic incompatibilities. Wolves and coyotes in North America are a classic example, and African savanna and forest elephant is another classic example. So these elephants are about 2 million years divergent, and yet where humans have driven the erosion of the forest savanna difference, really, if you like, forest elephants have been displaced by savanna elephants through the genome. So there's good evidence now that pretty much wherever there was forest, the invasion of savanna elephants went through, or at least was associated with increased gene flow between these two species. Now, so an interesting question, maybe important question, I would argue to answer, and would be what fraction of species diversity is it that's actually vulnerable to this process? I'm not aware of anyone having tried to estimate this. I think it's probably not very trivial to estimate that. So a couple of years ago, we've done some very crude back of the envelope calculations for just a couple of fish groups and a couple of mammals. And so these are the fractions that we estimated. So roughly 40% of these teleosts here have a relative that is less than 2 million years divergent, and 88% would have a relative that is less than 5 million years divergent. And these are the numbers for mammals. So it just suggests that if these figures are somewhat indicative of what these would look like on a larger scale, larger taxonomic scale, suggests there's a very large number of species that's vulnerable to this process. And of course, this may be particularly likely where species originated in response to divergent ecological requirements, divergent natural selection. And anything that would change the balance between divergent selection or disruptive selection and gene flow in these systems would potentially lead to the meltdown of reproductive isolation between them. One nice example is a little radiation of fish. This is sticklebugs in Enos Lake, work by Eric Taylor and others in Vancouver, where this is the classical species pair that Don MacPhail and Dorf Schlüter and others have been working with, or one of those pairs been working with for many years. And these were very distinct until somewhere probably in the end of the 80s or early 90s, and then they collapsed. And this was associated with a major change in the environment. I'm not going into the detail of this here. Now, we've just heard in the previous talk that hybridization can also be a source of diversity. There are many examples, and it's an increasingly large number of examples for hybrid speciation, like Nicola was saying. We actually see this also in the cichlids. Many of these examples, though, seem to be associated with the colonization of novel environments. And maybe sort of the first nice example, the oldest that I can sort of think of, is the Hawaiian silver sword radiation. So all of these species of silver swords in the Hawaiian archipelago, they have emerged from a hybrid population between two North American tarweeds that colonized the islands. So when can we actually try and make some predictions about when hybridization adds diversity and when it collapses diversity? So maybe not very trivial. And so we've done some very simple modeling some years ago without any genetic or ecological mechanisms. So it's really just a neutral model of biodiversity where we this was work done by my postdoc Gaku Takimoto and myself. So what we tried here is we used a simple neutral model of species diversity dynamics where change in richness through time depends on the intrinsic speciation rate of a clade, the number of species already present in the system, the extrinsic extinction rate, and the probability of establishment of a new species that was a function of the number of species already present in the system. And then we added this hybridization term. So now we allowed every species in the system to hybridize and make potentially hybrid populations. And we again used a probability of this hybrid species to either establish as a third species or replace one of the parents, or actually lead to the collapse of both parents as a function of how many species were already in the system. And it's almost trivial what you get out with such a model, but maybe it still helps to train our intuition about when hybridization might add positively or negatively to diversity. So what we found is that the effect of hybridization here on the richness of species depends on two things. It depends on the background rates of speciation and extinction in a lineage, and it depends on the heterogeneity in the environment, of course. So the number of niches that is effectively available. We found that in equilibrium, whenever speciation rates exceed extinction rates, so that's intrinsic speciation rates in the absence of hybridization, then adding hybridization, so here's the relationship between numbers of species in equilibrium and the number of niches in the environment as a function of hybridization, the rate of producing hybrid populations from nil to fairly high. So adding hybridization only decreases species richness. So it's intuitively obvious because speciation rates are sufficiently high for equilibrium species diversity to be be as high as it could possibly be. On the other hand, whenever speciation and extinction rates are about equal or extinction rates exceed speciation rates, we find that adding this hybridization term increases species richness, equilibrium species richness. So one example perhaps where intrinsic speciation rates are exceedingly high would be the cichletfish in the African Great Lakes. So here's just a slide with lots of different species of Lake Victoria cichlets. You can find up to 80 species in a single community. And a long time ago, this is how I got interested into this effect of environmental change on speciation reversal. I tried to understand the relationship between the number of species that can coexist in a community and that they are mostly isolated by having different signaling systems, or males signal different types of colors in particular microenvironments where particular light conditions prevail, and females mostly use these male color signals. Females don't have the colors, just the males have. The females use this information to choose their mates. And the lake has seen major loss of species richness in recent decades, and it has seen two major changes. One is the invasion of a large predator, and the other one is eutrophication, which changed the light environment from something that looked like a broad ambient light spectrum here. These are different light spectra measured at 2-meter water depth in a clear water place where you have all these different colors of the rainbow visible. And in the murky water place, there's almost only reddish and yellowish available. And we were able to show back then that this width of the transmission spectrum predicts almost entirely the number of species that can coexist in a community within any one genus of these cichlids. So, we still work on that, but I'm not going to show you any more work on cichlids today because I think it's perhaps interesting to move closer to the Mediterranean here. So this is not quite the Mediterranean, but it's Switzerland, and actually partly is Mediterranean drainage. And I think some of the work we've done recently here speaks maybe to the generality of some of the processes that I showed previously with cichlids, but also speaks to issues related to Mediterranean region biota. So Switzerland is extremely diverse in terms of its freshwater biodiversity because it's naturally connected to all the seas of Europe pretty much, and all these biotic components meet in Switzerland, or potentially meet, and certainly recently met because people helped meeting. So there's a unique archipelago of large, very deep, very cold lakes, both south and north of the Alps. And in fact, very many cold-adapted fish that have really small endemic ranges in Europe occur in this region. So we've been working for a couple of years now on one group. It's whitefish. They are related to trouts. And there used to be at least 28 species that we know of and that we have historical data for in Switzerland in 1950. And 58 years later, we only find 18 of these species and quite a bit of extinction. We've been interested in trying to understand what mechanisms were invoked in this extinction process. We had reasons to expect that it was not just demographic decline, but speciation reversal. I should say we were quite lucky that this system, we could do this work in a system because we had exceptionally good historical data. There was a fantastic natural historian who wrote a monograph on the Swiss whitefish 60 years ago. Something that's very rarely available for European fish, actually. And also there was very good historical fish collection that this person left us behind. And there was very good scale collections that we could actually extract DNA from to test the idea that species were lost through hybridization. And we could also infer, then it would help us to help inferring the mechanisms was also just possible because we also studied the ecological processes and the evolutionary history of this whole diversity of whitefish at the same time. So here's a phylogenetic tree based on some 1,000 AFLP loci for all the species that we could still get now of Swiss lakes. And all it shows you is that effectively every lake had its own radiation. And these were very recent because the lakes didn't exist until the ice retreated. And the species are all endemic to these lakes now. So in terms of the ecological mechanisms that structure these communities genetically and phenotypically, I just want to show you one slide and want to tell you just that water depth and feeding ecology are two dimensions of the environment that are important in structuring that. And here's a slide where I just plot, these are individuals all collected in one lake, and I just plot their score on the first CVA axis of shape against their score on the first FCA axis of microsatellite genotypes. And you see that these seem to be quite well related, and the relationship is actually mediated by water depth. So here's fish we collected at 2-meter depth, 15-meter depth, 30-meter depth, 60-meter depth, and 90-meter depth. So depth seems really important in structuring these. So then we were able to, gathering data on historical records of species across all these lakes, we found that the historical species richness was actually reasonably well explained or predicted by the maximum depth of these lakes. And some of these lakes are very deep, like we're talking of 300 meters and even more. Then came a major change to the environment, is eutrophication. Like you probably know, many of the Central European lakes, and I guess also the Iberian lakes, underwent eutrophication in the last century. Here's just the total phosphate concentration in the water. Note that there's a logarithmic scale. Each of these lines is one lake, one of the large deep lakes. And the bold one here is actually Lake Constance. And I'll show you a bit more data about that lake. And so you see all of these lakes became really eutrophic, really polluted. And then many of these have become clean again. Some have not yet. There's a few lakes that have moderate pollution only. So first up here show you that the percentage of species loss from nil to complete is quite well explained by the maximum pollution level that we know that these lakes had. And now the question is this perhaps because, well, I forgot to tell you that eutrophication leads to the oxygen depletion in deep waters of these lakes. That's because of the increased primary production and the decomposition of this material at great depth of the lakes. And so the lakes have become completely anoxic, many of them have, in deep waters. And deep waters are needed by these whitefish to breed, to hatch their eggs. So demographic decline could have caused this. But then we find that among those lakes that still have species diversity, that subset of lakes here, we find that the global genetic differentiations, the global FST between species within any one lake is a very steep negative fit to the maximum pollution that there was. And then we had, for Lake Constance, we were in a very lucky situation to have had access to historical scale collections that were not very nicely cleaned. So fisheries biologists tend to clean scales very well. It's very terrible for people that want to do genetics on them, because then there's no tissue, no muzzle left to get DNA out of. But we had access to a scale collection that was not very well cleaned, and so we could get DNA out of them. And so we did for Lake Constance. We compared genetic differentiation between these 4 species that are in the system through time. So here's each bar is one species contrast. They were all very highly, significantly differentiated prior to eutrophication. And here's what happened. During eutrophication, they all came down, and they actually continued coming down. And one pair is barely significantly differentiated now. So I think this is evidence for collapse of these species through hybridization. There is a 5th species in the system, or was in this system. It's extinct. It's been extinct for at least 30, 40 years. And we had all scales for that too, and we found several of its original private alleles now segregating in the other species. So additional evidence that also this species merged into this current hybrid swarm. So in a nutshell, this is what happened. So 15,000 years ago, these lakes were colonized, and adaptive radiations happened in these lakes, and and significant functional diversity has evolved that within just the last 50 years has been lost very quickly through loss of the dimensionality of breeding habitat. Important to see this is not so much the dimensionality of feeding habitat, because most of these species feed relatively shallow, either in open water or inshore, but they segregate during breeding. Some go really deep and others breed shallow. And this is what has been lost. So they have been forced to breed at the same depth, or those that breed deeper wouldn't be successfully recruiting. And those genotypes among them that would breed shallower and nearer to other species would have been favored during the eutrophication. So then we often ask, or we ask ourselves, and others ask us, so does it actually matter? I mean, these are relatively young species. They will probably re-evolve. So one quick answer is yes, they will re-evolve, I would think, but it won't happen at a time scale that is relevant to our society and not our children or grandchildren. We actually look at that in some of these lakes that have been restored to really good conditions after the collapse, and these fish have not recolonized the deep waters. So this is not a thing that happens immediately. I actually think it will take probably quite a while. But are there ecosystem or food web consequences? So until recently, there's one paper by people in Dorf Schlietter's group that tested whether diversification in an adaptive radiation as a stickleback affects ecosystem functioning. And they showed that when you have a sympatric species pair in mesocosms, you get different communities of zooplankton, benthos, zooplankton in different light conditions than when you have either one of these specialists alone or generalists. It was quite interesting. But this study didn't differentiate between evolutionary effects and plasticity, because they used fish taken from the wild. So we wanted to do this again, slightly differently, and we took 2 types of whitefish, 2 species from the same lake. We bred them in the lab, and then we raised them in the reciprocal feeding conditions, and then we measured the feeding performance and also their ecosystem impact in little model toy ecosystems, if you like. So here we show that genetic effects on benthic feeding. So here we measured the time to food depletion when they were forced to feed on benthic food. Has a very distinct genetic effect. So here's the large benthic raised on benthic food, and here's the little pelagic raised on benthic food, and here are the 2 raised on limnetic food. You also see immediately that there's also a phenotypic effect, but there's a genetic effect too. And interestingly, the largest effect is when you have the combination of the right genotype and the right environment. So this is here the benthic fish fed on benthic food, and the limnatic genotype fed on limnatic food. They're tremendously different in their efficiency of feeding. We see similar differences when they are forced to feed on plankton. So here the difference go in the other direction. So the little limnatics are significantly better at filtering plankton than large benthics. So there's good reasons to think that if you would now have them, have ecosystems that have either both a benthic and a plankton feeding specialist, versus an ecosystem that has just one generalist, there may actually be differences in how these ecosystems, how the food web is structured, or how energy is flowing through the food web. So we tested this in little mesocosms. And these are really toy ecosystems. We'd love to do this in real lakes, but currently we don't have the means to do that. So in these little toy mesocosms, we actually found very strong effects of phenotype. So depending on what you measure, so here doesn't so much matter, but here's the number of snails in the system, the number of Dreissena clams, the macro zooplankton diversity, and something else. What is it? I can't read it. It's the primary production. You get very strong effects of phenotype, and sometimes these are mediated by phenotypic plasticity, like here, when these two things have the same trend, and sometimes by genotype, like here, and sometimes the genotype that has been raised on the contrasting food regime is intermediate. So clearly these fish affect these little ecosystems in different ways. We still don't know from that now whether it matters to large ecosystems, but we think there's probably it's not unreasonable to think that it may matter, because these are the most abundant fish in the pristine lakes, at least in these lakes. And these lakes are not very species rich. So remember, this is sort of the classical diversity function relationship, and these post-glacial lakes are probably somewhere here, if not even further down here. So really, it might be that they have a fairly steep relationship. We'd expect a steep relationship between diversity and ecosystem function. Okay, so now I'll just change to a few slightly more speculative slides. This is on work that has recently started and currently ongoing. And it's about genetic effects of transalpine range expansion, particularly from the Atlantic, range expansions from north of the Alps to south of the Alps. There's also some in the other direction. And so I'm making an argument here for that there's probably a couple of cases that look like reproductive isolation meltdown between sympatric species, like the one I just showed you for whitefish and I told you about for cichlids, that these can be mediated or facilitated by the arrival of an unrelated allopatric relative. And the best probably example for this is trouts. Those of you that are familiar with trouts know that there's just one type of trout north of the Alps in pretty much all of continental Europe. It's a very widely distributed taxon. There's very little genetic structure across wide range of northwest Europe. Very different in the south. So northern Italy has several distinct species that are coexisting in sympatry. This is the famous marble trout, and this is the Adriatic brown trout. Here's some now already very old phylogeographic work that confirms that these are very distinct taxa. Here's the distribution patterns. This is from an old paper by Louis Bernatchez. And so these are really fully sympatric. This is completely nested within this. And here's a little endemic that is fully sympatric with both of these just in a single lake. So, and in fact, it's to just bring back to connect to the theme of the previous talk, that hybridization can generate diversity, and not only colors diversity. This is true also for fish, like someone asked this question here. Jennifer, you asked this question. And in fact, one of these trouts, this one, that's endemic to this big, deep lake, Lake Garda, has recently, well, now it's already a while ago, has been shown by Giuffra et al. To actually be of hybrid origin post-glacial hybrid origin between these two species, has invaded a totally new niche. So these are both river trout, and this is a lake trout that lives at 200-meter depth in this very peculiar lake. But more lately, this other trout, which has this wide northwest European distribution here, the northern brown trout, has been transplanted by people and has been stocked in very large quantities into these rivers. And Geoffroy et al. Were the first to actually suggest, on the basis of data that they had collected back then, this was allozyme data and mitochondrial sequence data, that the reproductive isolation between these historically sympatric, relatively distantly related sister species is eroding due to the arrival of the Atlantic allopatric species that seems to act as a bridge for gene flow between the two sympatric species. We've only recently started working on this system, and so now 20 years after Geoffroy, we actually find that these two species seem to be, I mean, we have a hard time finding any genetic structure, any neutral genetic structure. So here's just one. This is just factorial correspondence analysis. So then you see that that's the structure, and similar approaches just don't find anything else than a single population. But when you use phenotypes to make your groups and calculate factorial correspondence scores, then you find that they are still somewhat different. So this is Cinerinus, this is Marmorata. And now this is the phenotype of the invasive northern lineage. And it seems like you see on this axis, it's really intermediate between the two, and it seems it goes right in there and in here. We're currently studying that system with RATAC data. I don't have the data yet, or at least I can't show you today, but it seems actually confirms what we saw previously with AFLPs and microsatellites, effectively complete breakdown of reproductive isolation between these two previously sympatric and fairly distinct species. Now, how common is this? So maybe this be a common problem to many of the Adriatic endemic freshwater fishes, and perhaps elsewhere too. So we've launched a regional project involving northern Italian colleagues, French colleagues, and Swiss, to assess species distributions of fish across 20 of these large and deep lakes. We actually think that these are unique ecosystems that have been heavily messed around with, and it's high sort of time to get some handle on what happens in these lakes. And I just show you one list of species here. This is Lago Maggiore, one of the most species-rich lakes. I think we have some 49 species of fish in there, and in this column is native species, and in the other column here is invasive species. So, and here I broke these down into invasive here and native here, just to give you an idea what the fraction of invasive species are in these systems. And this is not one of the worst lakes. And then here's the fraction among the invasives of the species that have a close and sufficiently close relative in Lago Maggiore that they can integrate with. And here's the number of indigenous species that are sufficiently close to one of the currently present invaders to merge. And if we were to add more species from the north, this fraction here would increase. So more of these species have relatives north of the Alps. Can't show you any more data yet, except that here's just some impressions. So this is a recent invasion that wasn't really documented at all. This is northern roach. It's super dominant, at least the phenotype is super dominant in some of the southern lakes, and it seems to have lost to the complete, apparently it was associated with a complete loss of a pair of indigenous species, Rutilus aula, the Triotto, and the Pigo, Rutilus pigus. And we fear that this may be a more common situation. We actually think it's also a very interesting problem from an evolutionary biology point of view because some of these transitions happen in the other direction too. And it seems we don't see this breakdown of species in the north. And this may have to do with the fact that these southern species may have, in the history of sympatry, they may have evolved reproductive isolation, whereas the northern species is a recently expanding after the retreat of the ice, northern species that is not have any recent history of sympatry with congenes. So perhaps it sort of mates almost randomly, and therefore when it arrives in the south, might behave like a bridge for gene flow. But it should also be said that these lakes undergo various other human-driven changes like eutrophication, so these lakes change quickly, and this may be, and these things interact, and it's hard to distinguish it was hard to tell whether this hybridization would have happened also in the absence of the perturbed state of the ecosystems. Now, last couple of slides, just moving really into the Mediterranean. Is there any reason to think that these mechanisms may actually apply to the marine systems? There's clearly a lot of evidence for invasions affecting marine biota. But if you combine this with hybridization, there's almost no studies. It's really, I didn't know this until a couple of days ago when I looked this up. So people that study invasion biology in the sea do not study, with a few exceptions, a few notable exceptions, do not study genetic interactions between species. So here's a paper that many of you may be familiar with, pointing out, it's a recent review on invasions in the Mediterranean Sea, pointing out that this is the most heavily invaded sea in the world. I forget the numbers, like 60 species or something that have invaded just within the last 100 years or so. And this is very interesting, again, from an evolutionary biology point of view, because this system didn't actually exist, for fish at least, until about 4 million years ago. So during Messinian salinity crisis, most of the marine fish went extinct. And the entire system just got recolonized at the end of the salinity crisis, somewhere 4 million years or so ago. And recolonization happened predominantly from the Atlantic. So that really means all the endemic diversity that has arisen since, and there is a lot of that, is sufficiently recent for the lack of complete post-zygotic isolation between sympatric species, like here's wrasses that have radiated within the Mediterranean, and also between the Mediterranean and their Atlantic sister taxa. And in fact, the system is being invaded from two sides now, but these two invasion fronts have very different evolutionary histories. So one is here through the Suez Canal from the Red Sea. And these are, without exception, as far as I know, distant relatives that are way beyond the window of opportunity for hybridization with Mediterranean species. And the second invasion comes from here. It's driven by range expansion of Northwest African marine Atlantic fish possibly due to climate warming, and spilling over into the Mediterranean as they move all the way up into Western Europe. And these are the close relatives. So it would be really very, very interesting system, I think, to study the differential impacts of invasions with genetic interactions and invasions without genetic interactions. And this is just to illustrate, this is an anecdote, and then I finish. This is an extremely dynamic system. So on Monday, just before coming here, I went down to Tarifa. I'd never been there before. I wanted to see this place, which is historically such an interesting place, but also from by a geographical point of view, because this is Morocco over there, and here's Spain, and this is where all the invasions from the Atlantic have historically happened and where they're happening now. Just to illustrate how dynamic this system is. So this is a species of breams that is very common in the Mediterranean Sea, and I surely expected to see it there. I did some snorkeling, and what I found is something that looks different. In fact, this is Diplotus praensis, which is a species that, according to my field guide, which is now admittedly already 10 years old, restricted to the Cape Verde Islands. So this is a highly dynamic system. So I think like probably every few years, a different Northwest African species arrives in the Mediterranean. So I think it's a really important and interesting system from both evolutionary biology and conservation biology point of view. A couple of conclusions. So hybridization will probably be common, or expected to be common during range expansions. It can have very different consequences for diversity of species, and its consequences may depend to some extent on background rates of speciation and extinction, and also on the state of the environment. And speciation reversal through changes in gene flow selection balance, I'm afraid, could be a lot more widespread than we typically think. The problem with detecting is that probably most often when you suspect it has happened, it's already too late. Because the process, like we saw in the whitefish and in the cichlids, happens within a decade or so. It's gone. Yeah, the phenotypic structure can be gone really quickly, and the genetic structure too. And then to show it then needs more sophisticated approaches. I suspect it would happen often very early on when pristine ecosystems are being impacted. And I suspect it has significant consequences for ecosystem functioning or food web functioning. On the more applied side or more practical side, I think recognizing and documenting the real species diversity in such systems will usually require application of population genomics to biodiversity discovery, but also to biodiversity monitoring. So like many of these closely related species will not easily be differentiable unless you have lots and lots of genomic loci to show. Take the example of Lake Victoria cichlids. People still ask me, are these real species? And when we had like a handful of microsatellites or a few hundred AFLPs, we could never show that species are really reciprocally monophyletic. Now that we have several tens of thousands of SNPs, we can actually show that, and they are completely reproductive. They're completely, sympatric species are completely monophyletic reciprocally, even though they're very recent. And finally, so I think preventing extinctions through this process will require that conservation biology, beyond being concerned about the particular taxa, that they become concerned about maintaining the processes that generate and maintain these species through genetic mechanisms. I'd like to just thank a couple of people. Over here is more like my cichlid research group, but in this talk, really, most of the data I showed were either Pascal van Lanten, he worked on the species reversal in whitefish, this was his PhD project. Ben Lundskard Hansen and Blake Matthews worked on these mesochasm effects, on the effects of speciation reversal on ecosystems. And then Guy is the guy who's leading this large project across alpine deep lakes. And Irene Keller did a lot of work and does a lot more work on the trout introgression. Thank you for your attention.